right now, somewhere off the coast of northern Australia, drifting through warm, shallow water that barely reaches your waist, there is something hunting. It has no brain, no bones, no blood. It is 95% water, almost completely invisible, and it carries enough venom to kill 60 adult humans. And here is the part that should genuinely unsettle you. If enough of its tentacles wrap around your leg or your arm, your heart can stop beating in under 2 minutes. Not 2 hours, not 20 minutes, 2 minutes. That is less time than it takes to boil a kettle, less time than it takes to tie your shoes and walk to the end of your driveway. 2 minutes, and the most sophisticated organ in your body, the one that has been beating without interruption since before you were born, simply shuts off. Just like that. This is Chironex fleckeri, the Australian box jellyfish, and it is widely regarded as the most venomous creature on the planet. Not the most venomous fish, not the most venomous marine animal, the most venomous creature, period, across every kingdom of life. Pound for pound, its venom is more lethal than that of any snake, any spider, any scorpion. And unlike those animals, it does not bite. It does not need to coil or strike or lunge. All it has to do is brush against you. And in that single, brief, barely noticeable instant of contact, millions of microscopic biological weapons fire into your skin faster than a bullet leaves a gun. That is not an exaggeration. Researchers at the University of Heidelberg measured the speed of a nematocyst discharge, the tiny stinging cell that delivers jellyfish venom, and found that it generates an acceleration of 5.4 million times the force of gravity and exerts a pressure of more than 7 billion pascals at its tip. That is on par with the force of certain bullets, and it all happens in less than 700 nanoseconds. For reference, a nanosecond is 1/billionth of a second. A human blink takes around 350 million nanoseconds. By the time your eyelid finishes one single blink, a nematocyst could fire 500,000 times over. So tonight, as you settle in and let the sound of the ocean carry you toward sleep, we are going to spend some time with this extraordinary, terrifying, and deeply misunderstood animal. We are going to trace its lineage back more than 500 million years, to a time before trees existed, before fish had jaws, before anything with a spine had yet crawled onto dry land. We are going to look at how this creature hunts, how it sees the world through 24 separate eyes despite having no brain to process those images, and how its venom unravels the human body at the molecular level. We are going to meet its tiny, almost invisible cousin, the urokanji, whose sting is so psychologically devastating that victims have begged their doctors to let them die. And we are going to learn exactly what to do if you or someone near you is ever stung. Because that knowledge, those few specific steps taken in the right order at the right time, can mean the difference between a terrible afternoon and a funeral. But we begin where all good stories begin, at the beginning. And in the case of the box jellyfish, the beginning is so ancient it makes the dinosaurs look like recent arrivals. The phylum Cnidaria, the great family tree to which all jellyfish, corals, and sea anemones belong, is staggeringly old. Cnidarians first appear in the fossil record roughly 600 million years ago, predating the Cambrian explosion by a comfortable margin. That makes them one of the oldest multi-organ animal groups on the planet. To put that number in some perspective, the dinosaurs went extinct 66 million years ago. The first recognizable primates appeared around 55 million years ago. Modern humans have been walking the Earth for roughly 300,000 years. Cnidarians have been floating, stinging, and pulsing through the world's oceans for a span of time so vast it nearly defies comprehension. They were here before the continents had finished separating. They were here before flowers existed. They have survived at least 5 mass extinction events, including the one that ended the reign of the dinosaurs. And they have done it all without a brain, without a heart, without centralized blood circulation, and without anything that most of us would recognize as a face. For decades, the fossil record for jellyfish specifically was sparse. Jellyfish are, after all, mostly water. When they die, they dissolve. They leave almost nothing behind. So while paleontologists could point to 600-million-year-old polyp fossils, the free-swimming, bell-shaped medusa form, the part we picture when someone says jellyfish, remained elusive for the deep past. That changed dramatically in 2007, when researchers from the University of Kansas described 4 types of remarkably well-preserved jellyfish fossils from the Middle Cambrian Margin Formation in Utah, roughly 505 million years old. These fossils were extraordinary. They showed detailed soft part anatomy, including trailing tentacles and the surfaces of the bell. Even more stunning, the ancient specimens already showed the same level of complexity found in modern jellyfish species. They were not primitive precursors. They were fully formed, sophisticated marine predators. Then, in 2023, the Royal Ontario Museum announced the discovery of Burgosomjusa phasmiformis, an even more remarkable find from the famous Burgess Shale in British Columbia. 182 specimens of this previously unknown species were unearthed, some more than 20 centimeters long, with a cube-shaped bell and more than 90 finger-like tentacles. These were, without question, large, swimming, predatory jellyfish. They were 505 million years old, and some of them bore a striking resemblance to modern cubozoans, the box jellyfish. The class Cubozoa, which contains all box jellyfish, was not formally separated from the class Scyphozoa, the so-called true jellyfish, until 1973. Scientists recognized that box jellyfish were fundamentally different from their dome-shaped relatives in ways that went far beyond the shape of their bell. They had a unique life cycle, lacking the strobilation process that true jellyfish use to bud off multiple young medusae from a single polyp. They had complex eyes. They had active, directional swimming. They had courtship behavior. They had venom so potent it seemed almost absurd for an animal without a backbone. The evolutionary history of Cubozoa specifically is harder to pin down because their soft bodies fossilize so poorly. But molecular studies, combined with the handful of fossils that do exist, tell a story of deep antiquity. A possible Cubozoan fossil called Anthrocomedjusa turnbully has been identified from the Upper Carboniferous period, roughly 300 million years ago. And another possible specimen, Quadrumegicina quadrata, comes from the Upper Jurassic. Between the fossil evidence and the molecular clock data, researchers believe that Cubozoans diverged from other cnidarian lineages hundreds of millions of years ago, possibly in the Precambrian, or early Cambrian. Their unique traits, the box-shaped bell, the sophisticated eyes, and the direct metamorphosis from polyp to medusa, represent significant evolutionary innovations that allowed them to fill a distinct predatory niche. The deep divergences between Atlantic and Indo-Pacific clades of box jellyfish suggest that the breakup of the ancient supercontinent Pangaea, which began around 200 million years ago, played a role in shaping their distribution. Because most box jellyfish species live in shallow, nearshore habitats and cannot cross the open ocean, the movement of tectonic plates would have physically separated populations, driving them along independent evolutionary paths for tens of millions of years. This ancient vicariance helps explain why box jellyfish species today are found scattered across the tropics and subtropics, from Australia to Japan to the Caribbean, but with significant genetic differences between geographically separated populations. What is perhaps most remarkable is that these animals refined their venom over this immense timescale into one of the most effective chemical weapons in the natural world. A cocktail of proteins so perfectly calibrated that it can shut down a vertebrate heart in moments, and they did it without ever developing a brain. Consider what that means in practical terms. The box jellyfish that swims through the shallow waters off cairns today is not fundamentally different in design from the cubozoan-like organisms that swam in the warm shallows of the Cambrian seas. The basic blueprint, a pulsing bell, trailing tentacles armed with stinging cells, a simple nervous system distributed across sensory clusters, has endured for an almost inconceivable length of time. Most animal lineages that existed 500 million years ago are long gone. The trilobites, those armored arthropods that dominated the Cambrian seafloor for 270 million years, vanished during the Permian extinction. The ammonites, the spiral-shelled cephalopods that thrived for 350 million years, disappeared alongside the dinosaurs. But the cnidarians persisted, and the cubozoans, tucked away in their tropical nearshore habitats, riding the slow continental conveyor belt as Pangaea broke apart and the modern oceans took shape, survived them all. There is something humbling about that kind of endurance. We tend to think of evolutionary success in terms of complexity, bigger brains, better tools, and more sophisticated communication. But the box jellyfish offers a radically different model of success. It has stayed almost achingly simple in its structure, while becoming astonishingly sophisticated in its chemistry and sensory biology. It never needed to develop a backbone, a skull, or a cortex. It needed to see, to swim, and to kill efficiently. And over 500 million years, it honed those three abilities to a degree that puts many far more anatomically complex animals to shame. If you have ever seen a photograph of a box jellyfish, the first thing you probably noticed was how eerily beautiful it is. The bell of Chironex fleckeri, its main body, is pale blue and faintly translucent, roughly the size of a basketball when fully grown, typically reaching about 16 centimeters across, but occasionally growing to 35 centimeters. Viewed from certain angles, it bears a passing resemblance to a human head or skull, a coincidence that feels a little too fitting for the world's most venomous animal. The bell is not round like a typical jellyfish. It is distinctly box-shaped, with 4 flat sides, giving the animal its common name. This cube-like geometry is not just cosmetic. The rim of the bell folds inward to create a structure called the velarium, a muscular shelf that narrows the opening at the base of the bell like a nozzle. When the jellyfish pulses its bell, this velarium focuses the water jet, allowing the animal to move with a power and precision that no other jellyfish can match. Box jellyfish can reach speeds of about 1.5 meters per second, fast enough to actively chase prey, navigate around obstacles, and, if necessary, outswim a wading human. From each of the 4 lower corners of the bell hangs a short, fleshy stalk called a pedalium, and from each pedalium trail up to 15 tentacles. That means a fully grown Chironex Fleckeri can have up to 60 tentacles streaming behind it as it swims. When the jellyfish is relaxed, these tentacles contract to around 15 centimeters long and about 5 millimeters wide. But when the animal is hunting, they thin out and extend dramatically, reaching lengths of up to 3 meters. At full extension, they are gossamer thin, nearly invisible in the water, and absolutely loaded with stinging cells. Each tentacle carries roughly 500,000 cnidocytes, which are the specialized cells that house the nematocysts, the microscopic weapon systems that deliver the venom. Multiply that by 60 tentacles, and you begin to understand the sheer scale of the threat. A single box jellyfish carries millions upon millions of these tiny biological harpoons, each one primed to fire at the slightest contact with the right chemical trigger. And that trigger is important. Box jellyfish nematocysts are not activated by simple touch, like stepping on a nail. They are activated by chemicals found on living skin, specifically by the proteins present on the surface of human flesh, fish scales, and crustacean shells. This is why wearing pantyhose, a full-body lycra suit, or a wetsuit provides effective protection. The stinging cells react to the chemistry of a living body, not to the physical pressure of contact. The nylon or lycra acts as a chemical barrier, and without that protein signal, the nematocysts simply do not fire. This discovery has saved countless lives in northern Australia, where stinger suits are now standard gear for swimmers during the box jellyfish season. But perhaps the most astonishing feature of the box jellyfish is not its tentacles, not its venom, and not its speed. It is its eyes. Box jellyfish possess 24 eyes, arranged in clusters of 6 on 4 sensory structures called ropalia, which hang from the sides of the bell. These are not simple, light-sensitive patches like you might find on a flatworm. 2 of the 6 eye types on each ropalium are genuine, image-forming, camera-style lens eyes, remarkably similar in structure to the eyes of vertebrates like humans and to the eyes of cephalopods like octopuses. They have a cornea, a lens, and a retina. The lens is even made of a crystalline protein, the same type of molecule used in our own eyes. These evolved completely independently from vertebrate eyes, making them a striking example of convergent evolution, where nature arrives at the same solution through entirely separate paths. The remaining 4 eye types are simpler. The pit eyes contain only one cell type and probably function as basic light meters, detecting brightness and nothing more. The slit eyes are more complex, with 4 cell types and an asymmetric structure that may provide spatial resolution in a single plane. Researchers believe that each eye type serves a specific purpose, creating a system of specialized visual channels rather than a single, multi-purpose visual organ. In 2011, a research team led by Anders Garm at the University of Copenhagen made a remarkable discovery about one of these eye types. They found that the upper lens eyes of the box jellyfish Tripedalia cystiphora are permanently oriented to look straight up, no matter how the rest of the jellyfish's body is positioned. A weighted crystal, called a statolith, hangs from each ropalium and acts like a biological gyroscope, keeping the eyes level. These upward-looking eyes peer through the water surface into the world above, using an optical phenomenon called Snell's window to compress the entire 180-degree terrestrial visual field into about 97 degrees. The researchers demonstrated that the jellyfish use this view to navigate by detecting the overhead canopy of mangrove trees, swimming toward or away from the dark shapes of vegetation visible above the waterline. When the canopy was obscured, the jellyfish became disoriented and swam randomly. This is visually guided navigation, a behavior that requires image formation, spatial processing, and directed movement, and it is being performed by an animal with no brain. Only a loose network of nerve cells distributed across 4 ropalial ganglia and a ring nerve. It challenges everything we thought we knew about the minimum neural architecture required for complex behavior. To make the picture even more extraordinary, a study published in 2023 in the journal Current Biology demonstrated that Caribbean box jellyfish can learn from past experience. Researchers showed that the jellyfish could be trained to associate faint visual cues with collisions, modifying their behavior in future encounters. They were forming new memories without a centralized brain, using the ropalial ganglia as distributed processing centers, each containing roughly 1,000 nerve cells. By comparison, a human brain contains around 100 billion, 24 eyes, 4 distributed brains with 1,000 neurons each. No central processor, and yet it navigates complex environments, hunts actively, avoids obstacles, and learns from its mistakes. This is an animal that has been quietly demolishing our assumptions about intelligence for millions of years. To understand how the box jellyfish kills, you have to understand what happens at the molecular level in the moments after a tentacle wraps around human skin. The process is fast, efficient, and when the dose is large enough, almost impossible to reverse. It starts with the nematocysts. When the cnidocytes on the tentacle detect the chemical signature of living skin, they fire. Each nematocyst is a microscopic capsule, pressurized to about 150 atmospheres, roughly 150 times the pressure inside a car tire. Inside that capsule sits a tightly coiled thread, tipped with a tiny harpoon called a stylet. When the capsule explodes open, the stylet launches outward with that almost incomprehensible acceleration of 5.4 million times the force of gravity. At the microscopic scale, the pressure at the tip of the stylet reaches around 7.7 billion pascals, enough to puncture crustacean shells, fish scales, and human skin with equal ease. The entire discharge, from the moment the cnidocyle is triggered to the moment the thread is fully reverted and the venom is flowing, takes less than 3 milliseconds. The initial explosive phase, when the capsule ruptures and the stylet punches through the target, happens in under 700 nanoseconds. That makes nematocyst discharge one of the fastest mechanical processes in all of biology. Faster than any muscle contraction, faster than any nerve impulse, faster than anything else a living cell has ever been documented doing. But the real nightmare is what happens next, once the venom is inside your body. Chironex Fleckeri venom is an extraordinarily complex mixture. Researchers have identified more than 170 toxic proteins in box jellyfish venom. But the key players belong to a small family of pore-forming toxins unique to cubozoans. The best studied of these are CFT-X-1 and CFT-X-2, large proteins in the 42 to 46 kilodalton range that belong to a class of molecules not found in any other animal group. These are, quite literally, toxins that exist nowhere else on Earth. Here is what they do. These pore-forming toxins bore holes in cell membranes. They bind to the surface of a cell, insert themselves into the lipid bilayer, and oligomerize, meaning they link together with other toxin molecules to form a ring-shaped pore, essentially drilling a tiny channel straight through the cell wall. Once those pores are open, the cell can no longer maintain its internal chemistry. Ions that are supposed to stay inside flood out. Ions that are supposed to stay outside rush in. The cell loses its structural integrity, and depending on the cell type, either dies or becomes functionally useless. When this happens to red blood cells, it is called hemolysis. The cells rupture, spilling their contents into the bloodstream. Among those contents is potassium, an ion that is normally kept at low concentrations in the blood because the body carefully sequesters it inside cells. When massive numbers of red blood cells rupture simultaneously, they release a flood of potassium into the plasma, a condition called hyperkalemia. And hyperkalemia is, in medical terms, an emergency. Potassium is one of the key ions that regulate the electrical rhythm of the heart. The carefully balanced dance between sodium and potassium ions across the membranes of cardiac cells is what generates the heartbeat. When the blood is suddenly saturated with excess potassium, that rhythm collapses. The heart's electrical system short circuits. The coordinated contraction that pumps blood through your body devolves into a chaotic, useless quivering, a state called fibrillation, which rapidly progresses to full cardiac arrest. That is one proposed mechanism. But the story may be even more complex. Researcher Jamie Seymour at James Cook University in Australia found that certain protein fractions in Chironex Fleckeri venom appear to attack human cardiac cells directly, causing rapid cell death independent of hemolysis. These type 1 toxins, CFT-X-1 and CFT-X-2, seem to have a specific affinity for vertebrate heart tissue, which may explain why Chironex Fleckeri is so much more dangerous to humans than other box jellyfish species, whose venoms contain only the less heart-specific type 2 toxins. The end result is the same. The venom creates a catastrophic cascade. Pain receptors scream, blood pressure spikes, and then plummets. Blood vessels go into spasm. The heart muscle, under direct chemical assault and simultaneously drowning in excess potassium, loses its ability to maintain rhythm. In severe envenomations, where 3 meters or more of tentacle have made skin contact. This entire process can unfold in 2 to 5 minutes. The lethal dose, 50, the amount of venom needed to kill 50% of test subjects in laboratory settings, is a staggeringly low 0.04 milligrams per kilogram of body weight. For comparison, the lethal dose 50 of inland taipan venom, one of the most toxic snake venoms on Earth, is 0.025 milligrams per kilogram by the intravenous route. Box jellyfish venom is in the same lethal neighborhood as the deadliest land snakes, and it delivers that venom through millions of simultaneously firing microscopic syringes that the victim often does not see coming. Since the first recorded death in 1883, Chironex Fleckeri has been confirmed to have killed at least 64 people in Australia alone, with some estimates placing the true number closer to 80, when historically misattributed drownings and cardiac events are included. Most of the recent fatalities have been children, whose smaller body mass means a proportionally larger venom dose from the same length of tentacle contact. A 14-year-old died from a sting in Queensland in February of 2022, and a 17-year-old died about 10 days after being stung at a beach on Queensland's Western Cape York in February of 2021. To understand where these encounters happen, you have to understand where the box jellyfish lives. Chironex fleckeri inhabits the warm, shallow coastal waters of northern Australia, ranging from Exmouth in Western Australia all the way around the top of the continent to Agnes Water in Queensland. They are most commonly found in water less than a meter or 2 deep, close to shore, often near river mouths, estuaries, and mangrove-lined channels. In Australia, the box jellyfish season typically runs from October through May, aligning with the warmer, wetter months of the tropical year, though stings have been recorded in every month except July. During the season, the animals drift into shallow bays and beaches, particularly after calm, warm days when the water is still and clear. But Chironex fleckeri is not exclusively an Australian animal. Its range extends northward through the waters of Papua New Guinea, Indonesia, East Timor, Cambodia, Malaysia, Singapore, the Philippines, and Vietnam. The closely related and equally dangerous Chironex yamaguchii was first described from Japan in 2009 and has also been documented in the Philippines. Throughout this vast Indo-Pacific range, box jellyfish pose a significant threat to coastal communities, fishermen, and tourists who may not be aware of the danger. The geography of risk is important here. In Australia, decades of public education, the installation of stinger nets at popular beaches, the placement of vinegar bottles at beach access points, the development of antivenom, and a robust emergency medical system have combined to dramatically reduce fatalities. Australia now averages roughly 1 box jellyfish death every 3 to 4 years, a remarkable achievement given the size of the threat. But translate that same animal to a coastline where none of those protections exist, and the picture changes completely. In the Philippines, an estimated 20 to 40 people die from box jellyfish stings every single year. And that number is almost certainly a significant undercount. Death certificates are not required in many Filipino coastal communities. Fatalities are often attributed to drowning, cardiac arrest, or unknown causes. Children playing in shallow water are stung and die before they can reach a hospital, and the event is never officially recorded. One researcher estimated that the actual number of annual deaths in the Philippines from box jellyfish could be in the hundreds. These are often the most vulnerable people in some of the poorest coastal communities on Earth, dying from an animal that most of them have never heard of. The Philippines has more than 36,000 kilometers of coastline, vast stretches of it within prime box jellyfish habitat. And until very recently, there was virtually no public education about the danger. In Thailand, at least 15 confirmed deaths from box jellyfish stings have been recorded since 2008, including tourists stung at popular beach destinations like Koh Samui and Koh Phangan. Malaysia, Indonesia, and other Southeast Asian nations face similar, largely undocumented challenges. A researcher who studies the issue estimates that box jellyfish have claimed more human lives over the past 40 years than sharks, and the gap between those numbers is not close. The difference is that shark attacks make international headlines, while box jellyfish deaths in rural fishing villages disappear without a trace. The life cycle of a box jellyfish is a study in transformation. Like all cnidarians, box jellyfish alternate between two fundamentally different body plans. The polyp, a tiny, sessile, vase-shaped form that attaches to hard surfaces, and the medusa, the free-swimming bell that we recognize as a jellyfish. But box jellyfish handle this transition differently from their true jellyfish relatives, and the details are fascinating. It begins with mating. Mature Chironex Fleckeri medusae breed during the warmer months, typically heading into the lower reaches of rivers and mangrove channels to spawn. The male releases sperm into the water, the female takes it up, and fertilization occurs. In some box jellyfish species, the process is far more intimate. The species Charybdia civicae, for instance, performs what scientists have called the wedding dance, a courtship ritual in which the male physically grabs one of the female's tentacles and tows her through the water, positioning her so that their manubria, their oral openings, are close together. He then passes her a sack of brightly pigmented red sperm, which she consumes, fertilizing her eggs internally. This is a level of behavioral complexity that you simply do not expect from a jellyfish. After fertilization, the embryos develop into tiny, ciliated planula larvae. These planulae swim freely for roughly 2 to 6 days before settling on a hard surface, perhaps the underside of a dock piling, a mangrove root, or a piece of broken shell. There, the planula metamorphoses into a polyp, a minuscule, pale, translucent organism with a mouth and a ring of tentacles. These polyps are extraordinarily hard to find in the wild. Very few natural colonies have ever been located, and most of what scientists know about box jellyfish polyps comes from laboratory-cultured specimens. The polyp is the hidden, patient phase of the box jellyfish life cycle. It feeds on plankton, grows slowly, and can reproduce asexually through a process called budding, creating copies of itself that expand the colony. In this form, the animal can persist for weeks or months, quietly waiting for the right environmental conditions. When those conditions arrive, something remarkable happens. Unlike true jellyfish polyps, which undergo strobilation, a process that slices the polyp into a stack of disc-shaped baby medusae like tiny pancakes. The box jellyfish polyp does something far more dramatic. The entire polyp body metamorphoses into a single juvenile medusa. The whole organism reshapes itself, developing the beginnings of a bell, sensory structures, and swimming muscles. It then detaches from its substrate and drifts away as a miniature, fully formed box jellyfish. One polyp becomes one medusa. It is a complete transformation, the Cnidarian equivalent of a caterpillar becoming a butterfly. Once free swimming, the juvenile medusa grows rapidly. Within weeks or months, depending on water temperature and food availability, it reaches full size and sexual maturity. A fully grown Chironex fleckeri is an active, visual, diurnal predator that hunts during daylight hours and rests on the ocean floor at night. Its diet consists primarily of small fish, prawns, shrimp, and other pelagic invertebrates. The jellyfish feeds by extending its tentacles as it swims, then pausing, turning upside down, and sinking slowly. Anything that brushes against the extended tentacles is instantly stung, paralyzed, and reeled in by the contracting petalia, which fold the prey toward the manubrium at the base of the bell. The speed of the kill is not just impressive, it is necessary. Box jellyfish prey on small fish, and fish are fast. If the venom took minutes to work, the prey would thrash free and damage the jellyfish's delicate tentacles in the process. The venom evolved to be instantaneous precisely because the box jellyfish, despite being a supremely efficient predator, is itself a fragile creature. It cannot afford a prolonged struggle. As for predators, the box jellyfish has remarkably few. Green sea turtles and leatherback sea turtles are essentially immune to the venom, their thick, leathery skin preventing the nematocysts from penetrating. These turtles eat box jellyfish with apparent relish. Certain species of fish, including butterfish, batfish, and rabbitfish, have also been observed feeding on box jellyfish, though the mechanism by which they avoid envenomation is not well understood. Blue swimmer crabs have been documented consuming tentacle fragments. Most adult box jellyfish appear to reproduce once and then die. A reproductive strategy called simulparity, seasonal blooms in northern Australian waters, where large numbers of box jellyfish appear seemingly out of nowhere in October and November, and then vanish by May, likely reflect synchronized spawning events. The adult medusae mate, release their offspring, and then die within days or weeks, their bodies dissolving back into the ocean that created them. The longest a box jellyfish has ever survived in captivity is approximately 9 months, and keeping them alive for even that long required the invention of specially designed cylindrical tanks in the 1990s. Before that, captive box jellyfish typically died within days, their delicate bells damaged by bumping into the flat walls of conventional aquariums. If Chironex fleckeri is the box jellyfish that kills with brute force, the urukanji is the one that kills with subtlety, and in some ways, it is even more terrifying. The urukanji jellyfish, most famously Carukia barnesi, is tiny. Its bell measures only about 1 to 2 centimeters across, roughly the size of a human thumbnail. Its 4 tentacles, though capable of extending up to a meter in length, are gossamer thin and nearly invisible in the water. Where Chironex fleckeri is a basketball-sized animal trailing 3-meter tentacles that leave unmistakable whip-like welts across the skin. The urokanji leaves almost no mark at all. Victims often describe the initial sting as feeling like nothing more than a mosquito bite, a minor annoyance easily dismissed. Many people do not even realize they have been stung, and then 5 to 40 minutes later, the syndrome begins. Urokanji syndrome is unlike anything else in the catalog of venomous animal injuries. It was first described in 1952 by Dr. Hugo Fleckeri, who named it after the Aboriginal Urokanji people living near Cairns in North Queensland, where stings were common. But the animal that caused it remained a mystery for over a decade, until 1964, when a toxinologist named Jack Barnes captured a tiny, unidentified box jellyfish near the site of a previous urokanji case. To prove it was the culprit, he did something that sounds genuinely insane by modern standards. He deliberately stung himself, his 9-year-old son, and a local lifeguard with the jellyfish. Within 20 minutes, all three were experiencing intense, boring back pain, followed by constant abdominal cramps. On the drive to the hospital, both adults began having difficulty breathing and cycling through spasms of uncontrollable vomiting. All three survived, but the experiment left no doubt about the connection. The syndrome that develops after an urokanji sting is systemic, meaning it affects the entire body, not just the sting site. Symptoms include severe, excruciating pain in the back, abdomen, chest, and limbs, profuse sweating, nausea and relentless vomiting, headaches, rapid heart rate, dangerously high blood pressure, muscle cramping, difficulty breathing, and perhaps most disturbingly, an overwhelming psychological sensation of impending doom. A feeling so intense and so specific that it has become one of the diagnostic hallmarks of the syndrome. Victims do not simply feel sick, they feel with absolute conviction that they are about to die. Some have begged their doctors to end their lives rather than continue enduring the pain. The venom appears to act as a presynaptic sodium channel agonist, triggering a massive release of catecholamines, the same stress hormones, adrenaline and noradrenaline, that flood your system during a fight or flight response. But instead of the brief, manageable surge you get from a sudden fright, the urokanji venom causes a prolonged, uncontrolled catecholamine storm. Blood pressure spikes to dangerous levels. The heart races uncontrollably. In severe cases, the extreme hypertension can cause pulmonary edema, where fluid floods the lungs, or even intracerebral hemorrhage, a stroke caused by a blood vessel bursting in the brain. 2 people in Australia have been officially confirmed to have died from urokanji stings. The true global number is certainly higher, but impossible to quantify, because the jellyfish is so small that it frequently goes undetected, and the symptoms, arriving with a delay and mimicking cardiac events, are easily misdiagnosed. How many heart attacks reported on tropical beaches were actually urokanji envenomations? Nobody knows. At least 14 different species of small, 4-tentacled box jellyfish are now known to cause urokanji syndrome or urokanji-like symptoms. Reports have come from Australia, Hawaii, Florida, the Caribbean, Papua New Guinea, and Southeast Asia. And here is the detail that keeps researchers up at night. The urokanji's range appears to be expanding southward along Australia's coastline, possibly driven by rising ocean temperatures. Stings that once occurred only in the far tropical north are now being reported as far south as Fraser Island in Queensland and Ningaloo Reef in Western Australia. These tiny, almost invisible, agonizingly painful jellyfish are moving into waters where millions more people swim. Unlike Chironex Fleckeri stings, which leave dramatic, ladder-like welts across the skin, Urokanji stings leave almost nothing, a small red patch, a goosebump-like texture, maybe a slightly raised area the size of the jellyfish's bell. Many stings happen inside the stinger nets that are designed to protect swimmers, because the Urokanji is small enough to pass through the mesh, which typically has openings of about 2 centimeters. Tourists who believe the nets make them safe are particularly vulnerable. If you are swimming in tropical waters and you begin to experience escalating back pain, abdominal cramps, nausea, and a strange, rising sense of dread within 30 minutes of getting out of the water, take it seriously. Get to a hospital immediately. Urokanji syndrome is treatable, but it requires medical intervention, typically intravenous pain medication, including opioids, aggressive blood pressure management, and cardiac monitoring. There is no antivenom. Treatment is entirely symptomatic and supportive. The body has to fight its way through the catecholamine storm, and the medical team's job is to keep everything running while it does. Now we come to what might be the most important section of this entire script. If you ever find yourself in a situation where you or someone near you has been stung by a box jellyfish, what you do in the next few minutes will matter enormously. Let us walk through it. First, get the victim out of the water. This sounds obvious, but it is the critical first step. Box jellyfish stings are extraordinarily painful, and the pain can be so sudden and so overwhelming that victims may lose the ability to swim. Drowning is a very real secondary risk. If the person is thrashing or struggling, approach carefully and get them to shallow water or onto the beach as quickly as possible. Second, call emergency services immediately. In Australia, that is 000. In the United States, 911. In whatever country you happen to be in, get professional medical help on the way without delay. Do not wait to see how bad the sting is. Box jellyfish envenomation can deteriorate from painful to fatal in minutes. And the only reliable treatments for severe stings, antivenom, cardiac monitoring, and resuscitation equipment, are available only at hospitals. Third, apply vinegar to the sting site. This is the single most important first aid step you can take. Vinegar, standard household vinegar, the kind you would use in a salad dressing, deactivates unfired nematocysts that are still clinging to the skin. Remember, when a tentacle makes contact, not every nematocyst fires on the first pass. Millions of these microscopic weapons may still be sitting on the victim's skin, primed and loaded, waiting to fire at the slightest disturbance. If you try to scrape them off, rub sand on them, rinse them with fresh water, or touch them with bare hands, you will trigger a massive secondary discharge that pumps even more venom into the victim. Vinegar prevents this by chemically neutralizing the trigger mechanism of the unfired nematocysts. Pour it liberally over the affected area for at least 30 seconds. In northern Australia, most tropical beaches stock vinegar bottles specifically for this purpose. Fourth, carefully remove any visible tentacles. After the vinegar has been applied, you can gently remove remaining tentacle fragments using tweezers, the edge of a credit card, or a gloved hand. Do not use bare fingers unless absolutely necessary. Tentacles that have been separated from the jellyfish's bell can still sting. Tentacles on a dead jellyfish can still sting. Nematocysts embedded in sand from a jellyfish that washed up on the beach days ago can still fire if you step on them. Treat all box jellyfish tissue as loaded weapons at all times. Fifth, manage pain. After the nematocysts have been deactivated and tentacles removed, applying a cold pack or ice wrapped in a dry cloth can help reduce pain. Some guidelines also recommend soaking the affected area in hot water between 40 and 45 degrees Celsius for 20 minutes. The evidence on heat versus cold is somewhat mixed and may depend on the species involved, but both approaches are considered reasonable for pain management while awaiting medical help. And sixth, the one that matters most of all, be prepared to perform CPR. If the sting is severe, if the victim was stung across a large area of their body, if they are a child, if they begin to show signs of cardiovascular distress, shortness of breath, confusion, loss of consciousness, or if they simply collapse, begin cardiopulmonary resuscitation immediately and continue it until emergency medical services arrive. In box jellyfish cardiac arrest, the heart has not been damaged beyond repair. It has been thrown into an electrical malfunction by the venom. If you can keep oxygenated blood circulating through manual chest compressions and rescue breathing, there is a chance the heart can be restarted by paramedics using defibrillation and medication. People have survived severe Chironex Fleckeri stings through sustained bystander CPR that bridged the gap between collapse and professional treatment. What you should absolutely not do: do not apply fresh water to the sting. Fresh water changes the osmotic balance across the nematocyst membranes and can trigger mass discharge. Do not rub the area. Do not apply urine, alcohol, methylated spirits, baking soda, lemon juice, or any other folk remedy. None of these have been shown to work. Some of them actively make the envenomation worse. Do not apply a pressure immobilization bandage, the kind used for snake bites. Pressure over box jellyfish tentacles causes more nematocysts to fire. In a hospital setting, treatment for severe box jellyfish stings includes intravenous antivenom. Box jellyfish antivenom manufactured by CSL Limited, which is a sheep-derived antibody preparation. It is most effective when administered early. For cardiac arrest secondary to box jellyfish envenomation, all immediately available antivenom, up to 6 ampules, may be given as a rapid intravenous push. Intravenous magnesium may be administered if there is no response to antivenom. Pain management typically involves titrated intravenous morphine. And then there is the cutting edge. In 2019, researchers at the University of Sydney, led by Associate Professor Greg Neely, announced a potential breakthrough: a drug that blocks the venom of Chironex Fleckeri. The drug, a cholesterol-targeting compound called HPBCD that is already considered safe for human use, was shown to prevent pain, tissue death, and scarring in mice for up to 15 minutes after exposure. The researchers believe the venom relies on cholesterol in cell membranes to gain entry into cells. And by pulling cholesterol out of the membrane, the drug blocks the venom's ability to form its lethal pores. As of the time of research for this script, the drug has not yet been tested against live stings in animals or used in human clinical trials. But it represents the first true molecular antidote for box jellyfish venom, a potential game changer for emergency treatment in remote coastal areas. It is worth pausing here to talk about what a box jellyfish sting actually feels like, because understanding the sensation can help you recognize it and respond quickly. Survivors universally describe the pain as immediate, overwhelming, and unlike anything they have experienced before. The most common comparison is being branded with a red-hot iron. One researcher, stung by just a single inch of tentacle that blew against his arm during a field collection, described it as one of the most painful experiences of his life, and said he could barely imagine what 100 times that pain would be like. The sting leaves distinctive marks on the skin, raised, red or purple welts in a cross-hatched, ladder-like pattern that follows the line of the tentacle where it made contact. These marks can blister, and in severe cases, the skin beneath them dies, a process called necrosis, leaving permanent scars that survivors carry for the rest of their lives. The whip-like, braided scars of a Chironex Fleckeri sting are immediately recognizable to any emergency physician who has worked in tropical Australia. Even if a victim survives the initial envenomation, complications can continue for days and weeks afterward. A severe, itching rash can develop in the sting area and persist for weeks. The skin may be left sensitized and painful. Some survivors report lingering symptoms consistent with a weakened immune response. The psychological trauma of the event can also be lasting, particularly for children and for the families of those who did not survive. Prevention, then, is worth every ounce of effort it takes. If you are swimming in tropical or subtropical waters where box jellyfish are known to occur, wear a full-body stinger suit or wetsuit. This simple barrier prevents the nematocysts from reaching your skin and eliminates the chemical trigger that causes them to fire. Swim only at beaches with stinger nets, and be aware that nets do not provide absolute protection, particularly against the tiny Urokanji species that can pass through the mesh. Pay attention to local warning signs and seasonal advisories. Avoid swimming at night when visibility is low and the risk of accidental contact is higher. And if you are in a remote area without medical facilities, carry a bottle of vinegar in your beach bag. It costs almost nothing, weighs almost nothing, and in the worst-case scenario, it could save a life. We have spent the last several thousand words talking about the box jellyfish as a danger, as a killer, as a medical emergency, and all of that is true and important. But as we bring this journey to a close, it is worth stepping back and considering the box jellyfish in a broader context, not just as a threat to human life, but as a creature that exists within a rapidly changing ecosystem, an animal whose story is deeply intertwined with the future of the ocean itself. Box jellyfish are not endangered. They are not threatened. In fact, if current trends continue, they may become one of the few groups of marine animals that actively benefit from climate change. Warmer ocean, temperatures favor jellyfish growth and reproduction. As waters that were once too cold for tropical species gradually warm, jellyfish, including potentially dangerous species, are expanding their ranges into new territory. The box jellyfish that were once confined to the tropics of northern Australia and the Indo-Pacific are slowly creeping southward and appearing in areas where they have not been seen before. That notorious box jellyfish sting season in North Queensland, which traditionally ran from roughly October through may be extending as warmer conditions persist for longer periods each year. Jellyfish, broadly, seem almost uniquely positioned to thrive in a degraded ocean. They can tolerate lower oxygen levels than most fish, surviving in dead zones where their competitors suffocate. They can tolerate the increased acidity caused by rising carbon dioxide levels. They reproduce prolifically. Their polyps are resilient, capable of going dormant and waiting out unfavorable conditions. Coastal construction of harbors, marinas, and artificial reefs provides more hard surfaces for polyps to colonize, accelerating reproduction. And overfishing removes both the fish that compete with jellyfish for food and the fish that prey on them. Some marine biologists have warned of a phenomenon called ocean jellification, a scenario in which overfished, overheated, oxygen-depleted ocean ecosystems shift from being dominated by fish to being dominated by jellyfish and other gelatinous zooplankton. A major study by researchers at the University of British Columbia examined 45 marine ecosystems around the world and found that jellyfish populations had increased in 62% of them and decreased in only 7%. Jellyfish are showing up earlier in the season and staying later. Blooms are becoming larger and more frequent. The mechanics behind this shift are worth understanding. Over the last century, average ocean surface temperatures have risen by roughly 0.9 degrees Celsius. That number may sound small, but its effects are enormous. Warmer water accelerates jellyfish metabolism and reproduction. Species that were once confined to narrow tropical bands can now survive and breed in waters that were previously just a few degrees too cold. At the same time, oxygen levels in the oceans have declined by approximately 2% over the past 50 years, a consequence of warming waters and nutrient pollution. Most fish struggle in low oxygen environments. Jellyfish, remarkably, do not. Their simple bodies and low metabolic demands allow them to persist in dead zones where their competitors and predators suffocate. Add overfishing to the equation, which removes both the species that compete with jellyfish for planktonic food and the species that prey on jellyfish, and you have a perfect storm of conditions favoring gelatinous dominance. Coastal development makes the problem worse in a way that most people do not consider. Every new dock piling, harbor wall, bridge foundation, and artificial reef provides a more hard surface for jellyfish polyps to colonize. Remember, the polyp is the hidden, asexual phase of the jellyfish life cycle, the tiny sessile stage that quietly buds off copies of itself, and when conditions are right, metamorphoses into free-swimming medusae. More surfaces mean more polyps. More polyps mean more jellyfish. It is an unintentional mass production system built into the infrastructure of modern coastal civilization. For box jellyfish specifically, This has direct implications for human safety. As their range expands and their seasons lengthen, more people will encounter these animals in the water. As coastal populations grow, more people will be swimming in areas where box jellyfish thrive, particularly in Southeast Asia, where education about the danger is often minimal and medical infrastructure may be hundreds of miles away. By the end of this century, an estimated 50% of the world's population will live within 60 miles of a coastline. More people in the water, more jellyfish in the water, and warmer water pushing dangerous species into new territory is a combination that virtually guarantees an increase in sting incidents globally. In the Philippines, where children die regularly from box jellyfish stings in rural coastal communities, the problem is acute and worsening. A Senate bill hearing in March of 2025 discussed enacting measures to prevent and manage jellyfish stings, driven by the deaths of children whose families had no idea the animals were in the water and no access to the basic knowledge of vinegar treatment. The Philippine Society of Allergy, Asthma, and Immunology recently released a jellyfish safety video as part of a new public awareness campaign. A small but meaningful step in a country where virtually no community-level education about box jellyfish had previously existed. Tragically, even as these awareness efforts began to roll out, 2 10-year-old children died from box jellyfish stings in separate incidents, underscoring the urgency of the problem. The challenges are not limited to the developing world. In Australia, monitoring and predicting box jellyfish movements is becoming more complex as the animals expand their seasonal window and occasionally appear in unusual locations. Environmental DNA, or eDNA, technology is being developed and tested as a tool for detecting box jellyfish presence in coastal waters without needing to physically spot the animals, which is notoriously difficult given their near-total transparency. Researchers collect water samples and filter them for genetic material shed by box jellyfish, then analyze the DNA for species-specific matches. If a match is found, it confirms the animals are or were recently present in the area. This technique works for both the free-swimming medusa stage and the bottom-dwelling polyp stage, giving scientists a far more complete picture of box jellyfish distribution than traditional visual surveys ever could. There is also the question of what role box jellyfish play in their ecosystems, a question that gets far less attention than the threat they pose to humans. Jellyfish are not empty calories drifting aimlessly through the ocean, as they were long assumed to be. They are active participants in marine food webs. Box jellyfish consume vast quantities of small fish, shrimp, and planktonic invertebrates, exerting top-down pressure on prey populations. Their bodies, though mostly water, contain carbon that gets transported to the deep ocean when they die and sink, contributing to the biological carbon pump that helps regulate atmospheric carbon dioxide levels. Their waste and shed mucus support microbial communities that play important roles in nutrient cycling, and they are, in turn, food for other animals. Sea turtles, certain fish species, and crabs all consume box jellyfish, passing that energy up the food chain. Removing box jellyfish from their ecosystems, or viewing them purely as threats, would be a mistake. They are, like every other organism, a product of their environment and a functional component of it. The problem is not that box jellyfish exist. The problem is that we are altering the oceans in ways that disproportionately favor jellyfish over fish. And we are putting more people into the water in the places where the most dangerous species live, without giving those people the information they need to protect themselves. The box jellyfish did not evolve to hurt humans. It evolved to catch shrimp and small fish in tropical shallows. Every death it causes is an accident, a collision between a transparent predator that cannot see a waiting child and a waiting child who cannot see a transparent predator. The venom exists because a soft-bodied animal with no bones, no claws, and no teeth needed a way to instantly immobilize fast-moving prey without getting its fragile tentacles torn apart in the struggle. It is one of evolution's most elegant and lethal solutions. A chemical weapon so refined that modern science still does not fully understand all of its mechanisms. There is an ongoing scientific debate about the precise mechanism by which box jellyfish venom kills humans. And it is worth noting because it illustrates just how much we still do not know. One camp, led by researcher Angel Yanagihara at the University of Hawaii, argues that the primary lethal mechanism is pore formation in red blood cells, leading to massive potassium release and hyperkalemic cardiac arrest. Her research has demonstrated this pathway convincingly in mice and has led to the development of a topical cream used by United States military divers. Another camp, led by Jamie Seymour at James Cook University, points to the direct cardiotoxic effects of specific venom proteins that attack heart cells independently of hemolysis. Seymour notes that autopsies of human sting victims show no signs of widespread hemolysis, suggesting that something other than potassium flooding is killing the heart. The truth may involve both mechanisms working simultaneously, or may vary depending on the dose and the individual victim's physiology. The fact that scientists who have studied this animal for decades still disagree on exactly how it kills should tell you something about the complexity of the challenge. What is not debated is the need for better preparation, better education, and better treatment options in the regions where box jellyfish claim the most lives. The tools exist. Vinegar works. Stinger suits work. CPR works. Antivenom works when administered early. What is lacking in too many coastal communities around the world is awareness that these tools are needed and access to the resources that provide them. Every box jellyfish fatality in a community that has never heard of vinegar treatment is a preventable death. Every child who dies on a beach that has no warning signs and no emergency medical access is a failure of communication, not a failure of science. And that, perhaps, is the part that stays with you. Not the fear, not the statistics, but the strangeness, the sheer improbability of this animal's existence. 500 million years of evolution, no brain, no bones, no blood, and yet it can outswim you, outsee you, and stop your heart in the time it takes to send a text message. It is older than the dinosaurs, older than flowers, older than trees. It has survived ice ages and asteroid impacts and the breakup of continents. And it is still out there, right now, pulsing through warm coastal water in the dark, transparent and weightless, and loaded with enough venom to kill a room full of people. The ocean is not ours. We are visitors there, wading into a world that was ancient before our species even existed. And the box jellyfish, silent and invisible and lethally beautiful, is a reminder that the oldest residents do not always welcome guests. It does not hate us. It does not fear us. It does not know we are there. It simply drifts and pulses and hunts, the same way it has hunted since long before the first fish grew legs and crawled onto dry land, since long before the first tree cast a shadow, since long before anything on this planet looked up at the stars and wondered what they were. 500 million years. No brain, no bones, and still the most venomous animal alive. Sleep well.